Okay, folks, let's go ahead and get started with our webinar today. All right, I'm seeing a lot of thumbs up. Thanks very much. So, I am Sam Moyer. I'm a research fellow here at NDI's Emerging Technologies Institute. And today we have a really interesting topic for you for our monthly Tech 101 webinar series. We have Dr. Richard Murray, who we'll introduce in a moment, and he's here to talk to us about synthetic biology. As synthetic biology rapidly evolves with things like advances in AI, automation, manufacturing, and a lot of other trends that Dr. Murray will speak to us about, these trends are really having a major impact on our economy and our national security. Similarly to how chip advancements have transformed computing, a lot of people think that synthetic biology is poised to transform biomanufacturing and the economy more broadly. In the future, biomanufacturing could become a key way that the US produces materials for both commercial and defense applications. We're talking about substitutes for commodities like fuel, but also innovative new materials that we might not even be able to imagine yet. So this is going to be a really interesting conversation delving into synthetic biology today. Some quick admin things. All of the attendee lines are muted, so you're muted. If you have questions, please ask them, and the right way to do that is to use the Q&A box, not the chat feature. So please use put questions in, and just know that we're holding questions till the end so we can accumulate them throughout the conversation, and then Richard will do his best to answer them at the end of the webinar. We are recording today's webinar for members who are unable to attend. And this webinar is part of a monthly series. And if you're interested in getting updates on future Tech 101s and our other webinars, please sign up for our newsletter, and maybe someone can drop a link or to our website where you can do that. So now to introduce our guest, Dr. Richard Murray was educated at the California Institute of Technology and University of California, Berkeley. He is currently the Thomas E. Doris Everhart Professor of Control and Dynamical Systems and Bioengineering at Caltech. His research focuses on the application of feedback control to network systems with applications to synthetic biology and other areas. And his current projects include the design and implementation of synthetic cells, as well as many other things. He's actually currently on sabbatical. He's living here in DC, and he's working in the overlap right now between biotechnology and US national security and strategy. So thank you very much, Richard, for joining us today. Over to you.
Great. Thanks, Sam. Let me share my slides so you guys can all see them. And Sam, if you can confirm for me that those are up. Everybody seeing those?
Yes, they're up.
Fantastic. Thank you. Great. Thanks to NDA for inviting me to speak, and Sam for that introduction. I want to go through a little bit of synthetic biology and why it matters for national security. There's a QR code up on the right-hand side there, and all of the slides I'm showing are available there, and links to more information. And so that's a linkable PDF. I'll put it up again at the end, just in case you don't catch it this time, or you can just send me an email, and I'm happy to send them to you. So I kind of got interested in synthetic biology now about 25 years ago. And from the point of view of an engineer coming from kind of robotics and autonomy and controls, thinking about biology as a programmable machine. And so, when you look at what biological systems can do, they do look a lot like machines. This is something called a neutrophil. It is chasing a bacterium in a field of red blood cells. I think we can all sort of see it is chasing. I mean, there's no question about it. But we can think about, what's the decision making that's going on? How is it sensing where that bacterium was? How is it deciding to change its shape? How is it then engulfing it, which is what it does? This is part of your immune system. And so I've been interested in thinking about synthetic biology from that point of view. How do we design complex machines? And so can we think of this like we would think of a computer or something, but at a micro scale? And so that's kind of the perspective that I bring to this. But of course, biotechnology is much broader than just that vision of kind of creating biology as machines and synthetic cells and other things that I'll touch on briefly. And so what I want to talk about today is first a quick introduction to synthetic biology, just to kind of level set everybody about some of what we're talking about, talk about some of the application areas that are in particular of interest to the defense sector, talk about some of what's going on internationally and what some of the competition looks like, and then give a little bit about my future perspectives from a kind of US biotechnology strategy point of view. All right, so let's start with a little bit about just, what is synthetic biology? So when we talk about synthetic biology, we start by mining things from nature. So we take natural organisms, and we look at the various proteins and other machinery that they have available and mine these into parts. And so you see here, an example of two parts. One is something called a repressor. This one sort of here on the upper side, where there's a protein. If it binds to the DNA, it can repress the expression of a gene, which would produce another protein. So we think of that as a switch, right? Something is switching something off. If it's not present, it allows it to be on. And you can have the opposite polarity. You can have something called an activator, where a protein binds to a piece of DNA and then turns on expression. So switch with the opposite polarity to it. And once we have these kind of basic building blocks, and there are many, many more, of course, hundreds of things that biology can do, but we can take these basic components and put them together in ways maybe that biology didn't put them together. So for example, we might think about making something like a ring oscillator. So that little symbol there is a repressor. And so we think of each of those circles as representing a protein or a gene encoding for a protein. If that protein is expressed, then it represses the next thing down the line where that little T-shaped symbol is pointed. And if you think about the logic of this for a little while, if the top thing at the very top is a protein and it's expressing, it will turn off the next protein, which will turn on the next one, which will turn off the next one, which will turn on the next one, which will finally turn off the one at the top, right? We started on, we go off. And so this is an oscillator, right? It's a ring oscillator, build this in electronics all the time. We can have that conceptual design, but then we can go and look at these parts that we've mined from nature, characterize them in terms of repression characteristics, and eventually pick the specific component. So here I'm showing a specific set of proteins by those little acronyms that are there that we put together. And this circuit is not something that exists in nature. First, there aren't ring oscillators so much in nature, but also these particular proteins are not in nature put together these ways, but we can rearrange the DNA, right, and conceptually put together a new circuit. So what we do is we do the design at that level, and then we have molecular techniques that allow us to go in and actually code up DNA that does that. And so we can essentially put those DNA sequences on something called a plasmid. You can do this with DNA synthesis. That costs 7 cents a base pair. This is maybe I don't know, $5,000 or 5,000 base pair long thing. So maybe $350 worth of kind of initial manufacturing. Put on a plasmid, and then replication costs are zero, right? So you think of this as I build, my stamp or whatever to make a bunch of these things, and I stamp them out. And here, once we have that DNA, we can put it inside of a living organism. And of course, living organisms see DNA and they execute whatever program is on that DNA. And in this case, they would execute that ring oscillator program if it works right. So there are techniques for doing all of these things. And synthetic biologists think about how do we implement interesting functions. And I'll give you a lot of other interesting functions in a little bit. But this one, as the ring oscillator is a good example. So we can then put this inside cells, and we've done that. And this is some work from one of my PhD students from about a decade back. What you're looking at on this picture on the right is something called a mother machine. It's a microfluidic device, and you see the cells there. They're in channels, and you'll see as I turn the movie on, they start growing, but they have this circuit embedded in it. And one of these proteins is bound to a fluorescent protein. And so what you're going to see is these things glow on and off. And in fact, if you look at this one that's about 5 down, you can see it's already starting to glow at the start of the movie. But when I run this, and you see the scale here, these things are a couple of microns long. So these are really micro scale machines executing my program. And this is what that looks like when it runs. And so in each of these channels, you see that program running, and you see it glow on, then off, then on, then off, and it's oscillating, right? And so it's oscillating through the circuit. And so this might be something that we use perhaps as a clock inside of something. We want to time something, we want to keep track of how many cycles happen before something else went on. Maybe we want to synchronize things according to this. That would be a standard use in electronics. So this is an example of what synthetic biologists do. And typically the way we do this, I think in say the aerospace industry, we also have some background, right? We would, build complex models and do things, and then, eventually run it through different test rigs at different scales, right? And build them up and test them, wind tunnels, et cetera, et cetera. Here, similar techniques, less model-based and more experimentally based, partly because it's pretty quick to do this stuff and it's pretty cheap. And so in the synthetic biology community, you'll hear this sort of design, build, test, learn, right? Design it, build it, try it out, see what happens, figure out why it didn't work, iterate, iterate, and that fast iteration allows us to get things working quickly. And you can think of this as, this is what biology does too, through mutation and evolution. We're just doing that in a more directed way. Okay, so that's sort of, my one slide overview of how synthetic biology works. And you don't need to, kind of understand much more detail than that to understand what I'm going to talk about. Okay, so what do people do with our ability to essentially insert arbitrary DNA and have it execute a program into cells? Well, there are a number of different applications in synthetic biology. The ones that are here now that you maybe already know things about are things like making chemicals, right? So biomanufacturing of chemicals, whether those are fuels, for example, sustainable aviation fuels or other things. I'm showing here a metabolic pathway that is a sequence of chemicals that are converted from one to another by enzymes that create something called 1,4-BDO, which is a precursor for a variety of plastics. You essentially put that into a big fermenter, and like you would brew beer, you now brew 1,4-BDO. So that's one big application area, biomanufacturing. I'll say a little bit more about that. Another certainly is crops, GM crops, genetically modified crops. And so, for example, we can engineer into the DNA of a corn plant something that allows the corn plant to resist different types of insects, right? Or resist different types of blight, or grow better, more uniform corn kernels, right? Whatever the trait is that we're trying to do. And in the US, this is, very common. Probably 95% of all corn that you eat and use in a variety of corn-based products is a genetically modified corn. We have super high yields, right? We're able to use less pesticide by virtue of building in the insect resistant into the plant itself. So agriculture in the US especially, not every place in the world, but many places in the world is another great area for synthetic biology where we're doing things. And then, of course, there's the whole medical side of things. So diagnostics and therapeutics for human health, whether those are monoclonal antibodies or different types of sensors. And I'll talk more about what that sensor picture is a little bit later. So these are here now. There are companies, they do these things, they make money off of them, or they don't, they go out of business. I mean, all of those things. Pretty mature area. Has been around, for the last, probably 50 years, if you go back to chemicals and insulin as the first sort of product there. What people are doing now is getting fancier about these things. And so we think about things like engineered microbiomes. Can I engineer microbes, living bacteria, that are part of a kind of fancy probiotic, right? So you're going to eat a pill, it's going to have bacteria in it, they're going to go colonize your gut. Look maybe for sources, they'll colonize different parts of the gut, and those that end up in places where there's inflammation, perhaps release a therapeutic ingredient or therapeutic chemical that helps relieve that inflammation, and does so in a very localized way, right? So it does it right where you need it. And you can think about microbiomes. There's your gut microbiome. There are all sorts of microbiomes on your skin. When you have a wound, there's a microbiome in that wound. Soil microbiomes, that's called the rhizosphere around a plant. Waste microbiomes, all of those we can think about as engineerable things if we engineer the microbes to do something, both individual microbes, but also consortium microbes. And that's an emerging area. Another is engineered living materials. I'll talk also more about this one, where we can think about, for example, engineering concrete that can repair itself, or engineering other types of materials that can repair themselves or react to different chemicals or react to different optical signals. So engineered living materials is another, and they don't have to be purely living. They can be hybrid devices, like hybrid silicon cell devices that are also coming out. And then there are the really long range applications and the types of things that we think about might be possible in the next 20 years, perhaps. And so, for example, I work in the area of engineered synthetic cells. Can we engineer from scratch, bottom up, cells that essentially implement the functions that biology brings us? And can we use that to engineer synthetic biomachines? So, for example, could I engineer, as sort of pictured there, something that was a machine made out of biological parts that extracted chemicals from the environment around it, transported them to processing centers to convert them to a different form, transferred them to packaging centers where it transforms them into some nice package. And of course, that's an orange tree. And I don't know how to build an orange tree as an engineer, but I'd like to. I think that's that where we're headed. And if I can build an orange tree, can I build a tree that sort of makes oranges and lemons and plums, right? Sort of all on the same tree. I think that's possible. I know you can do it with oranges. And can I do so in which the root system is adapted to different environments and other things? So think about synthetic biology as, can we engineer that plant? And it might look like biology does it, start with a seed and have it grow, or it might be something that we just 3D print, and we 3D print a machine that does that in some way. And then you can think about engineering ecosystems, right? Can we engineer insects, for example? And people are, starting to look at these things, or do conservation, right? And so maybe some of you have heard about the dire wolf, right? So you think about what can we engineer in terms of traits and other things that sort of benefit the overall ecosystem that we live in on the planet. So these are the types of things that that people talk about. Again, the ones on the far left are here now. They're companies. The ones in the middle are things are forming. The ones on the right-hand side are where researchers like myself at Caltech spend our time thinking about, wow, what could we do? Okay, so let me dig into a couple of these to kind of talk about, both what's currently being done and how it's being done, give you a little bit more sense of what synthetic biology can do, but also what some of the challenges are and maybe some of the overlaps with national security policy. So let's start with biomanufacturing, that kind of upper left one, if you will. And so, of course, biomanufacturing to some extent is very well understood, right? Beer is biomanufacturing, right? I mean, it's natural microbes as opposed to engineered microbes, but we basically treat the cells as a chemical factory. We essentially, convert sugar into alcohol. And so we know how to do that well, and there, sort of fermenters all around the world making beer. Probably, sort of every single geographic region probably has fermenters that are making alcohol. And we can think about that, sort of in a more modern sense about saying, well, what if we want to make something besides alcohol? What if, for example, we want to make insulin or vanillin or artemisinic acid, which is a precursor to artemisinin, which is a malaria drug. And so you can think about doing that by instead of inserting repressors and activators, like I showed you on that kind of first slide, what if I put in enzymes that are specific to taking some source, some substrate, carbon at its basic level, and producing some product, whether that product's a bulk chemical like fuels, a fine chemical, pharmaceuticals, whatever it is. And of course, what we do is we put in DNA that encodes for enzymes. So, DNA goes to mRNA through transcription and then through translation to enzymes. And we get to choose what the enzymes are by virtue of the DNA that we put in. And again, we can order DNA now for 7 cents a base pair. We can put it onto a plasmid. We can drop it in to E. coli or yeast. And have this metabolic pathway, enzyme 1 to 2 to 3 to 4, that produces that product. And so those sorts of metabolic engineering techniques really open up what we can do. One example of that, and sort of the last one on my list there, is artemisinin. Here is the pathway to go from a simple sugar to artemisinic acid, which is a precursor via a chemical process to artemisinin. And as I mentioned, artemisinin is a malaria drug. Normally, we get artemisinin from plants. That is, this pathway exists in plants. We harvest the plants, we extract out the artemisinic acid or the artemisinin, depending on how we're doing it, and that's what we do. The problem is if there's a breakout of malaria, what are you going to do? Go plant the plant and grow it and then do it? It's a year before you have the drug. If I can take things that are normally in plants and put them into yeast, right? I just need to turn on a fermenter, right? And so the next day I can have some artemisinic acid, and maybe within a week I can have useful quantities of artemisinin sort of flowing out. And this kind of basic idea of taking metabolic pathways from animals and plants, so insulin, right, coming from your body or pigs, right, and if you're diabetic, put that into E. coli or yeast. Artemisinic acid, let's put that from plants into E. coli or yeast. And so it provides a method for taking things that we can biomanufacture and put them in. And so I mentioned, for example, 1,4-BDO as a commercial product. This exists. There's a company called Geno that builds this product. It's a precursor for a lot of different kind of bioplastics, plastics, I suppose I should say, and then this is a biological technique to get there. And I'm just showing a part of that pathway, but it's basically that same idea. So that's something we can do, right? And of course, I think what's interesting about it is that not only can we do things that nature already does, like artemisinin or different types of alcohols, but we can also think about new materials, new energetic materials that maybe have different properties, new synthetic rubbers, novel materials that we've never seen before in nature. And Francis Arnold, who won the Nobel Prize for direct evolution, likes to say, nature is the best chemist there is. And partly what she means by this, proteins can do molecular modifications. And if we can design the protein to do the modifications that we want to the molecules that we want, we can make really new things. So we can make new proteins to make new chemicals with novel properties. And of course, this is of interest to every different field, but certainly in defense, if you're thinking about things like energetics, fuels, rubbers, these things that matter a lot to what's going on, and where the properties make a big difference. Now, there are a lot of challenges to doing this right. Scaling up, going from something at the lab scale of maybe sort of 100 microliters to 1 to 10 liters that you might still do in the lab, to getting up to kiloliters of sort of capacity. So I'm showing here on the right, something that's a large scale biomanufacturing facility, and you've got these big tanks, and in these tanks, right, are these bugs that are doing this thing. It's hard in part because what happens in the lab may not match what happens in the factory. It can be expensive to build that, sort of facility that you're looking at there down on the right, and that's a bunch of CapEx costs. You're competing with the traditional chemical industry in many cases, so for things like fuel and rubber, and they're hard to beat, right? I mean, they're really good at it. They've invested all their CapEx and amortized that, and so that can be very tricky. It's also an international market, and other places have different prices, both for the substrates, but also for the labor. And of course, sometimes when you make these biomanufactured materials, it might have better properties, but it might also be different. And so you can't just drop it in, for example, without modifying or at least retesting what's going on. So I think it's a challenge there. It's a challenge I think that in principle we are up to and addressing. I will say this particular plant that I'm showing you over there is actually in China, not in the United States. And China has been investing big in doing this. The US has in the past as well, but it's an area, as I'll say a little bit later, that we're falling behind a little bit in. Okay, so that's an application area that currently exists. Another one that I mentioned was doing types of diagnostics and therapeutics in the kind of health space. And so in that area, sort of looking maybe a little bit more forward, right? We can think about traditional analytical instruments. This is a sort of mass spec from like an undergraduate laboratory. They can get smaller and more modern, but it's nonetheless it's a big machine. And this may not be the type of thing you want to take out in the field if you're a soldier or you're special forces or something. And, these are just not particularly portable, but they are very sensitive. And a question is, can we do things similarly in biology? Biology can detect chemicals. I mean, that's what happens when you have a reaction to something, for example. And there are a lot of really interesting technologies that are coming along. One that I quite like, and as an example, are paper-based assays. But also whole cell sensors and other things. So I promised to say a little bit about this picture up on the right. This is something that Keith Party did when he was a graduate student working with Jim Collins, who's now at MIT. And this is a little paper-based piece of paper with each of those spots having a synthetic circuit in it that detects different strains of Ebola. So this is now a DNA level, right? Is it this strain or that strain? It shows up on the DNA sequence. And what color it turns tells you what it's finding. But you can imagine not only looking at DNA sequences, but also small molecules, right? All sorts of things. Some of those things we can do in traditional ways, right? Just the chemical strips that we see for like COVID tests. But other things, like which strain of Ebola is it, is not something you can do. But this is now something, it's a freeze-dried paper assay. You basically, spot it with water that rehydrates a synthetic circuit that goes and says, if it's, Ebola type A, turn green. If it's Ebola type B, turn red. And then you can point your cell phone at it, right? And you can see you can have a bunch of different spots. They can tell you different things. They can give you different concentrations and other things. There are also things that people have done with recording, not just statically, did I see something or not, but also dynamically. Did I see one thing followed by another thing? What order did they happen? How long were they separated in time? So recording events and keeping track of properties of those events. This technique of cell-free synthetic biology is a very interesting one. It's being picked up within DoD. This is an example of a device using that same technology that you see above that was done by the Chemical Biological Center as part of the Army, DevCom. And so here you're looking at something that now they've kind of encapsulated this in a little device. You can see the size of it there. Somebody's holding it in their fingers. And again, you drop water on this. It's got a positive control. So it's a lot like a COVID test. In fact, I think this one is actually, they're using it, COVID as their example, but you can detect many other things besides just is there COVID, do you have COVID or not, but rather what type, what else is there, or what other small molecules did I see? And so I think this sensing area is one that's very interesting. It's sort of happening now. People are doing this. And there's a lot on the horizon. I think it's a great area for synthetic biology. So as we get increased sensitivity, complexity, the ability not just to sense, but also to respond, right? The number of signals that we can handle. Again, these things about complex ordering of events, temporal ordering of events that we might encode in the DNA so that you see what this cell saw and when and for how long, and it gets encoded in the DNA, and there are ways to do that using things called integrases. And then you can kind of read it out on a sequencer afterwards and said, okay, this is what you were sort of exposed to. And then how do we think about not just detecting those things, but doing something about it, right? So synthesizing maybe a chemical. I mentioned the gut microbiome examples. That sensor might trigger something that releases a therapeutic locally in the region you need it. One of my favorite applications that just came out actually, maybe a couple months ago, was some work that Chris Voigt's group did at MIT. One of his postdocs, now research scientist named Jonathan Chemla. And this was a bacterial sensor that is a hyperspectral imager. So I'm just showing the kind of output of this, but they basically encoded bacteria that could detect different types of chemical in the environment and then essentially synthesize chemicals that have an absorption spectrum so that you can see how they reflect back just plain old light. And what they've done here is in a field setting, they've spread bacteria, and in these two little regions that you see here, they put different chemicals, and the bacteria respond to those chemicals. And then they image this from, I can't remember if it was 100 meters or 50 meters. They basically take a drone with a hyperspectral imager, fly over this, right? and then take that data back and do a lot of analysis on it, and they can see those two spots. This one's hard to see the sort of stuff picking up, but here you can kind of see it. And it says, hey, in that region, the following chemical is there. Maybe that's an explosive, maybe that's a signature or something that you wanted to see. And you can see it's very sensitive, right? And again, this is just flying over it in a drone and measuring that. And so I guess here, not flying over it, but 90 meters away, right? They're sort of measuring this just from a post, right? So, but really, impressive, and you can think of lots of potential applications. Okay, so in this sensing and diagnostics area, again, another great application area, lots going on. And I mentioned engineered living materials is something that's more coming. And so, I think the thing to remember here is that biology has an amazing set of things it can do with materials. And those materials can have chemical properties, mechanical properties, optical properties, thermal properties, all at once. And so just as an example of that, we take this example here. So that octopus, right, just decided, okay, and no longer need to hide out, right, sort of sitting, in this kind of coral or plant or whatever it is, right, I think I'll make myself visible. So what did the skin do to do that, right? I mean, pretty amazing thing. And of course, that same skin is feeding the octopus, is protecting the octopus, is providing thermal insulation. So we know biology can do these amazing things. And so how do we take advantage of that, right? For adaptive response, it has capacity to self-repair. How do we really do that? So this is an area I think that's more emerging as people do that. There are lots of case studies and examples. I've written some down here that are engineered materials, sometimes living, sometimes not. Spider silk, biocemented soil, bioconcrete, photonic materials, different types of textiles. And it really is, I think, an exciting area. An example now from maybe 5-6 years back is in this biocemented soil. So here they're spraying bacteria on top of soil, and then that bacteria excretes something that stabilizes that soil and increases the ground strength of that soil, so that maybe you can drive over it or land a plane on it or other things. As just one example. People have done things with self-healing concrete. So the bioconcrete example, this is an example coming in work there from a couple of years back, sort of showing that if a break occurs, you can sort of have that break get repaired by having something precipitate out calcite that then kind of heals it in some way. All right, so all of these I think are interesting examples. And again, as I mentioned at the start, these don't all have to be things that are just biological. We can think about things that are also hybrid, silicon and cells combined together, right? Or biomimetic versus biological. We take the principles of them, but we implement them in different ways. So lots of challenges with this. How do you get them to last long enough? How do you control it, make it robust? How do you fabricate it? How do you integrate it? All of those things. All right, so this gives you, I hope, a little bit of a sense about why this is an exciting area, including from the point of view of the defense industry and some of the things that people might do. So where are things at? And so let me say a little bit about kind of the international context and kind of plow into some of the maybe more national security related things. All right, so if we look at the international outlook, there's basically three big players here, right? The US, the UK, and China. I should say there are more, but I'm going to pick these three as good examples. And the US was the early leader, right? So recombinant DNA, insulin back in the 1970s, that's really where this whole revolution started in some sense. And there was an early vision for that. This is a study that came out of the DARPA Information Science and Technology Study Group, so ISAT, and ISAT did a synthetic biology study. I was part of that study. That's what got me into this field. And we had a roadmap for what was going on, right? We sort of said, look, we're going to be able to intentionally engineered cells. You see that sort of sitting in the middle of this chart. And there's a bunch of things that go along with that in order to get the design tools and the measurement tools and everything else to sort of work in the right way. But you see that sort of went out as the bar was in 2013. And I think one of the realities is that this has not gone quite as fast as we would like. And I think that's an interesting thing to think about. Nonetheless, there's a pretty rich ecosystem. This is back from 2017, showing just some of the companies that form this in areas like healthcare, industrial chemicals, biofuels, various synthesis techniques that you need to do that, agriculture, consumer products. You'll recognize some of these companies. In the upper left, you see Moderna as an example, right? They build a synthetic biology product, right? mRNA vaccines are essentially a simple synthetic biology product, but of that kind of class. So there's a great environment for doing startups. Some of these companies don't still exist, right? This is 8 years old. Many of them still do. And are doing exciting things in that world. And that's one of the, as we know, the strengths of the US environment. So another thing that is good about the US is that we think about what that roadmap looks like. And so this is something put together by the Engineering Biology Research Consortium. It's a roadmap. You see that design, build, test, learn cycle sitting down there at the bottom. You see some of the kind of component elements of synthetic biology, biomolecular engineering, host engineering, data science, engineering DNA, and then some of the application areas around that outside. The industrial biotechnology I've talked to you a little bit about, health and medicine, but there are others as well, like biomining, remediation, lots of different things. And if you're interested, the EBRC roadmap is a great place to look to see what's kind of coming and what's out there. So there have also been a number of national academies reports looking at the future of biotechnology. The Preparing for Future Products of Biotechnology in 2017 is one that I chaired, looking at the regulatory system and how to have the regulatory system be ready. Safeguarding the Bioeconomy is an excellent report talking about some of the things we need to do in the US to safeguard the bioeconomy. And perhaps the most recent, not out of the national academies, but out of Congress, is the National Security Commission on Emerging Biotechnology, which came essentially out of kind of chips and science and the Trump-Biden EO, and a sequence of things that happened, sort of going back, sort of 5 and more years. And the NSCB report, if you've not read it, I think is a great description both of what the opportunities are, what the challenges are, and what the US needs to do to get there. Having said this, for all of the wonderful things going on, the reality in the US is that outside of pharma, the biomanufacturing and to some extent synthetic biology community is really struggling on the industry side. So this is the stock price of an unnamed biotechnology company, biomanufacturing company. But you see this pattern a lot, right? So it is flat for a while, right? So this is going back to 2022. They open, the fact that it's $10 tells you it's probably a SPAC. And then they kind of dip and maybe come up a little bit, but all of a sudden they drop all the way down. I don't know if you can read that number down there, but I think it's probably like, 17 cents a share or something like that right now, right? And yeah, 17.85 a share. I guess we can see it up at the top, or 16.97. And so, if you were one of those $10 investors, you just lost a fortune. And unfortunately, this is not uncommon. This is just what it looks like. And I'll, say a little bit more about why that is, as we kind of go along. Okay, so that's sort of what's happening in the US. In the UK, they've been very good, and they were a very early player in what was going on. And so way back in 2000, which is sort of the start of the modern synthetic biology era, as people started to say, hey, we can really do this kind of at scale, they were there, and they were doing things. There's a program called iGEM, the International Genetically Engineered Machines Competition. They were there from the very beginning. It started in about 2004, and mainly US based, and they kind of came in very quickly. And then a whole sequence of things as you kind of go down that chart. The kind of leading, some of the leading groups there, Imperial College London's a big leader in that area. Sinby City in London is a kind of startup environment for that. University of Edinburgh also has a very strong group. And the UK is very good at international engagement, engaging with countries all over the world to sort of do that. The areas that they're looking at, pharma is one, right? And so you think about, the various pharmaceutical industries that are based in the UK, and there are quite a few. Maybe other areas as well. I think this is still evolving. Is, agriculture going to be a good? That's one for the US. It is different now, right? Since Brexit, they're decoupled from the kind of GMO debate. And so that changes things around a little bit. Okay, and then, of course, third is China. So China participated in a 6 academy study. That was the United States, the UK, and China, National Academies of Science and Engineering. So that was the 6 academy study. They've had a very aggressive set of directives as part of their 5-year plans. They have a different dynamic, right? So they import petroleum and food, right? We're a net exporter of both petroleum and food. And so we can make things out of petroleum, right, that China has to import the petroleum. And so they're very interested in alternative pathways to making different types of chemicals where, just like we're worried about being reliant on other countries, they are too. And so they have a different set of incentives for going forward. And similarly with food, they import a lot of food. They're great at investing in engineering. And of course, they have different societal norms, right? A centrally directed engineering mandate, that works differently in China than it does in the US. And this is from the NSCB report, one, kind of worrisome trend there is if you look at the number of top cited papers in synthetic biology. So these are papers that are in the top 10% of the most cited papers. China is overtaking, right, the US. So China is the top curve, the US is the next curve down in a very significant way. And this is not total number of papers. So this is not just kick out a bunch of papers, right? These are papers that are getting cited by others. And maybe there's some self-citations and other things in there, but this is a big enough difference that, we ought to be thinking about that. And they are also tracking what we're doing. So for example, that engineering biology research consortium roadmap was translated in Chinese about 2 years after it came out. And now the new ones that are coming out get translated almost immediately with help of LLMs and other things. So this is an area in which there's substantial competition, and China's playing to win, and they're doing a great job. So I think that's, my sort of view of what it looks like. I want to just say a little bit more about China. I showed you, sort of this plot. Another plot that, we can look at and be a little bit worried about is just this is now all of STEM, not synthetic biology, but annual STEM PhD graduates. And again, you see China going all the way back to the early 2000s, started to clip the US, both in terms of the total, including non-citizens, but also in terms of our domestic PhD production. And they're now, at whatever that is, 3.5 times what the US is in terms of our domestic education at the PhD level. Of course, China's a big country, 4 or more times bigger than the United States. And so maybe they've not caught up on a per capita basis, but this is one where just numbers can matter, right? They have a tremendous workforce that's available to them, and you see the rate of increase that's happening there. So again, these are things that the NSCB was very concerned about. When you go look at what's actually happening, though, everybody's worried about we're importing everything from China. 70% of our drugs come from China. Well, not quite. So, there are different measures of that. But if you look at API, or active pharmaceutical ingredient manufacturing facilities, only about 13% of those are in China. This may not be the right measure, right? Because the question is where are the materials for those factories coming from? But the US and the EU combined have a much bigger share of those manufacturing facilities. Similarly, this is just a different chart showing the same thing. If you look at the total regional capacity in North America and Europe are the two biggest, and China is sort of growing. This is a 2021 report out of BPI. The link's up there on the upper right. But, and it's hard to get these data, right? So what does it look like in 2025? I'm sure that bar has increased quite a lot. Okay, so, but as I said, they've got a strategy. They're investing in it. They may not have that kind of ecosystem of startups and other things yet, but they've got a lot of talent, PhD, both bachelor's all the way up through PhD, and they're really building what they're doing in manufacturing and also ag tech, right? And those are the two starting points for being the leader in biotechnology, is having a strong biomanufacturing and ag tech base. And so we'll see, sort of where that goes. And the US may be putting its leadership at risk, right? We have a lot of regulations and other things that get in the way. And I'm serving as a fellow at the National Security Commission on Emerging Biotechnology, trying to help with that regulatory side of things based on that report that I mentioned on future products of biotechnology. And of course, the commercial markets may not be enough on their own, right? The reality is that it's hard to beat the chemical industry. The reality is that it's hard to beat that international market. And so if there's not a commercial market for you, you just don't do it. And so we need to think about what to do. And again, the National Security Commission on Emerging Biotechnology said we need to invest, we need to coordinate, we need to streamline, we need to partner. And again, I encourage you to read that report if you haven't already. Okay, so just a couple of last things, and then I'll sort of pause and we'll have time for questions. I think one good question is to say, what's different about synthetic biology versus other emerging technologies? And I think there are a couple of things. Certainly one, this is true in other fields as well, but I think it's very much true in synthetic biology, is this convergence, democratization, and trust, right? So convergence, synthetic biology combined with AI, you've all heard of AlphaFold and other techniques, automation, cloud labs, self-driving labs, the ability to do these things automatically. Democratization, this stuff is now happening not just in large .coms and .edus, but smaller .coms, right, startups, but also, right, community organizations and, just sort of DIY bio labs, right? And the tools are getting easier and easier to use. And it's becoming increasingly something where you can just say, hey, I'd like to set up a small synthetic biology lab in my garage. Can I do that? And the answer is pretty much yes, you can do that. I mean, it's not that expensive and it's not that complicated. And generally, there's a little bit of a lack of trust, right, of what's going on in the public, Gen Y and Gen Z, international. A lot of our stuff's funded by the Department of Defense, so people worry about that. My top concerns for synthetic biology is a field, right? Thinking about this from a US perspective, right? But just as a field around the world. So one is this inability to scale. All of the things that we've built so far are kind of saturated out at a pretty simple scale. Things like this 1,4-BDO pathway might have a half dozen enzymes in them. The most complicated things that I've seen have a few dozen enzymes in them. The type of scale we need to do something like this neutrophil swimming through and detecting things, et cetera, that's got hundreds, if not thousands of of components. They're not engineered, they come from biology. But if I were to build that, think about building a self-driving car. This is a self-driving neutrophil in this particular case. Think about how to do that. So that's one. I mentioned already this kind of lack of commercial viability, especially in manufacturing. Here's another chart. This is for a company called Gevo. Again, you see that same pattern, right? Sort of starts up high and then just, tanks out, right? And is now trading at $1.18 a share after opening at, whatever, a very large number when you look at the K after those dollar values, right? So, I mean, it really is, just tanked. And this is, again, 5 different companies I looked at, they all have this sort of a pattern to them. Short-term versus long-term vision. I do think the biomanufacturing and ag are the right place to start, but the opportunity is much larger. If we want to build things like that neutrophil or synthetic plants or other things, I think we have to think bigger than just, kind of biomanufacturing and agriculture. And so again, we can start there, but if we want to get to something like, can I build a machine that can make, oranges, lemons, and plums all on the same tree in any part of the world I want, depending on what root system I order, that I think is going to be a little bit harder than what we're doing so far. And just generally, there's a kind of lack of understanding of biotech solutions. Many of you maybe are like me. I, took biology in high school, didn't like it, and that's why I became an engineer. Right? I was an electrical engineer by training, and didn't come back to kind of biological engineering until I'd been at Caltech for 15 years. Right? And so and now that's most of what my group does. And of course, there's no global ecosystem for this. The standards, the norms, societal, societies, technical societies. There's no international association of synthetic biologists, right? It's a little bit more split up than that. Okay, and then a lot of concerns just for what's going on in the US. I do worry that we have the risk of falling behind in synthetic biology leadership. The NSCB gave us 3 years. The regulatory complexity is high, so there are a lot of processes for doing that, and I won't go through these here, but if you're interested, I can talk about them in the Q&A. We have to think about releasing things into the environment, and so there are things called engineered microbes for environmental release that you might be doing for doing biomining or different types of environmental remediation. You put live living organisms in the environment, they will persist in the environment, they will spread. We need to think about that and understand that. We see that in other domains, things like CRISPR babies, invasive species. So this is an area I think that we should be worrying about. And supply chain dependencies, right? Sort of taking advantage of the biotech solutions. You got to build them up ahead of time, right? If somebody all of a sudden, like today, you probably read in the paper, right? China's, putting restrictions on critical materials, right? Critical minerals, right? Well, some of those can be biomined, but you got to build up that base to do that. People talk about generative AI and the concern of that. There was a nice National Academies report on that. I'm happy to talk more about that. If you know a little bit about mirror life, maybe you've heard that as well. I'm not too worried about that. Okay, so I'll maybe stop there and just kind of wrap up. So here again is that link. I think my main messages for you, and then happy to discuss these more in Q&A, are one, synthetic biology hasn't yet lived up to its promise. The yet is important. I think we can get there, but we really haven't cracked the code for engineering biology at scale. There are bright spots in pharma. Agriculture is doing pretty well in the US, right? But industrial chemicals and biomanufacturing is struggling and is, running the risk of going out of business, right? And again, here's one of those plots, different company, right? But there's a third one I showed you with that same pattern, right? Going down to zero very quickly. It's trading at $7 after a 40-to-1 reverse stock split, right? And so, that just kind of tells you the challenges in that area. At the same time, biology can do these amazing things, right? And so I showed you the neutrophil, I showed you the octopus, right? I mean, it's, incredible what it can do. And so, how do we latch onto that, right? And do those same incredible things as engineers. I think to do that, we need to create the engineering discipline that can create biological systems at scale. And that's in blue because it is what I'm spending my time here in DC doing, is trying to articulate a little bit about what that vision might look like. Maybe it doesn't quite look like the picture you see over there on the right, but something like that. And I think that's an important thing to do. And I do think it's possible, but it's not going to happen on its own, right? It needs vision, strategy, investment. We need to go beyond just biomanufacturing and agriculture, right? And really build what we need to have an innovation ecosystem in this area. And finally, if our country wants to get there first or be one of the first to get there, right, we're going to need to run faster, right? We're going to need to invest, coordinate, streamline, and partner. And again, that was the main message coming from the National Security Commission on Emerging Biotechnology. Okay, so with that, maybe I'll pause there and throw it back to Sam. I'll leave the slide up a little bit just in case you want to catch that QR code. But Sam, I'll throw it back to you for reading any of the questions you got or throwing in your own or whatever you'd like to do next.
Yeah, that's great. Thank you so much, Richard. We've got some good questions coming in here, and we've got up till the top of the hour to get through those. Please keep adding questions if you have them. So a couple questions in here about the industrial side that you've touched on. One of them talks about the point you made with the commercialization of this technology often involving high CapEx expenditures. That seems to fit the pattern that you were showing potentially with some of those companies and that struggle to commercialize. So can you talk a little bit about that? Is that the way you see it? And do you have any opinions on whether there's an opportunity there for public-private partnerships that can potentially help there, or what other kind of opportunities do you see to assist with that scale-up piece?
Yeah, that's great. So let me first, I'll summarize what I think, part of the challenge is, and then I'll, come to this, and what might we do about it. So Doug Friedman, who's the CEO for Biomade, right, which is a manufacturing institute, DoD manufacturing institute, likes to say, when you talk about the bioeconomy, right, so biomanufacturing is part of the bioeconomy, remember that the noun is the economy, right, and it's the economics of this that are really kind of getting in the way. There's the technology's more or less there. It can always be better. If it's better, we'll make more money, all those things. But the economics of it are not there. And I like to think of it as there are kind of two pieces, cost and price, right? So can biotechnology, can biomanufacturing beat the cost of other technologies? And so you've got the substrate costs, you've got the capex costs, and of course the chemical industry has all of that capex is sort of already amortized, and they're just investing in a continual basis. And a lot of things are sort of subsidized, right? They're subsidized because maybe we don't charge for sort of pollution, or we don't charge for carbon release or other things. Where biology might be better, it might have a lower cost, but if you're not giving monetary incentive back to that, then you can't realize that. And so one is just, can you produce at cost compared to other industries? And the other is price. And the reason I differentiate them is there's your cost to do this, let's say in the United States versus traditional manufacturing, but there's also the price that your competitors are willing to sell at, right? And in particular, in an international market, right, that cost can be subsidized, right? We subsidize, for example, agriculture, right? And so the cost that you can sell, right, agriculture outside the US is subsidized, right? And so similarly, other countries can subsidize their biomanufacturing industry, and that means that the price that you pay for that material might be higher. So I think that's part of the challenge. Can you get the cost down, and can you deal with the price? And the pricing, right, is where you start thinking about things like tariffs and other things, right, as a way to equalize that if it's really being subsidized. But to flip it to the question you asked, Sam, I think that now you can say, well, where could the US invest? Certainly you could do things like help subsidize the capex costs, right? So either, right, sort of give tax incentives, or do things where you build up the facilities so that early stage companies, right, don't have to spend that capex. They can just spend some amount that you've invested the capex. Some of that's happening, right? So the DBIMP program that the Department of Defense had, it's currently on hold, was building up some of those facilities in areas that the Department of Defense cared about. Biomade is building up some of those facilities. These are mainly early stage, not full scale up to production. But again, the US, like we're doing in the semiconductor industry, right, could say, yeah, why don't we do something similar in biomanufacturing? And the National Security Commission on Emerging Biotechnology recommended such things, right, that we needed to do that. Tricky to do, right, in a year when we're, doing tax cuts and other things. So we have to face the political realities of choosing, right, where we're going to do those investments, and that's something the country has to decide. Public-private partnerships can do a lot toward that, right? And so, for example, in Biomade, these are things where you can get company investments, you can get state investments, you can get federal investments, and combine them. So I think that's an absolutely great thing to look at, and getting the industry to sort of support their part of all of that to help build the overall industry, but including their own business space.
That's great. So I'm going to cheat a little bit. I work for NDIA. We're a trade association, of course. We had a question come in about workforce considerations. A lot of what you were describing, really fascinating, but also maybe outside the wheelhouse of
Yep.
a lot of folks who work in national security, but clearly massive implications for the future of national security. So I guess maybe I'll make it a two-in-one question. One is, do you think there's an opportunity for some potential workforce internal to the national security apparatus? Is there an opportunity to educate the workforce more, or do you think that's an important thing? And then the other piece of it is, you mentioned trade associations and the lack of associations for this particular domain. So, is there an opportunity for us who work in the association space to potentially do more to bring different communities together? What do you think's what's the low-hanging fruit there, in your opinion?
Yeah, that's a great question. One of the reasons that I was, excited about, giving this seminar was exactly kind of reaching out and doing just a little bit of, kind of bioliteracy. What can be done? Where can things go? Right? Because if you're like me, right, sort of, Sam, you mentioned you worked actually, right, sort of on the biotech side for a while, but most of us didn't, right? For me, it was that ugly stuff that I didn't like in high school. And I think it's different now. And I think it's more important now in a certain sense. I mean, it's always important for human health, but I think for other applications, it's now more important. And I think we need to get that word out, right? We need to educate ourselves. We need to educate Congress and the staffers on the Hill and other things. And those are areas where trade associations can play a big role, right? By bringing people together, doing things like you guys are already doing. There's a biotech caucus that the House is forming. It's got 18 members so far. They will be having, their usual information sessions and other things, right? And those are both opportunities to learn, but also opportunities to, help inform. So I do think that there are opportunities to do that. And I'd say for anybody, I'm here in DC doing these sorts of things. And so if you have connection to some community, say, boy, it'd be great to get Richard in here to help us stimulate a conversation about that. Feel free to send me a note. I'm happy to do that. I do think that it's important to talk about what can be done and to give people a sense. Um, in the, in the broadly biotechnology space, Biotechnology Innovation Organization, used to represent one of the many trade organizations that try to represent biotechnology. They decided now maybe about 4 months back to really focus on medical and pharma, right? And so they said, we're not going to do bioindustrial, we're not going to do agriculture, we're not going to do environmental. And so they've, really moved out of that area. There's something called the American Alliance for Biomanufacturing, AAB. And so if you go to aabio.org, probably, you'll find out more about them. They are one trade organization that's doing that. There doesn't have to just be one. There could be different ones, right? They are not particularly defense oriented, for example. So there may be opportunities for more. I personally, Richard Murray, thinks that we need a robust set probably of industry organizations that are really advocating for what needs to be done, right? That are going and saying, look, this is important for national security. This is important for our economy. This is what we can do when bills come up. So there are a half dozen bills right now sitting in Congress that are coming out of both the NSEB report and other things. And we need companies in the states where they have representatives saying this is jobs for our state, right? You need to look at that. You need to support that, right? You need to join the House Biotech Caucus if that's important for the representatives in your district. So I think just companies going out, and again, trade organizations will organize those things. They put together Hill Days, right? They'll do all of that stuff. So I think it's important with bio moving out of this space. I think there's an opportunity. Bioindustrials, AAB is one place that can pick some of that up. Maybe it's not everything. It's got to figure out, what its focus is brand new. What are we going to do on the ag side of things, right? What are we going to do on the biomining side of things, right? Because biomining is different, right? Those are environmentally released, right? That's, I think, for the most part, AAB is probably going to focus more on fermentation and bioindustrials, right? And it's different. And biomining, if you're trying to get critical minerals and other things out of waste products or other things, that's an area that needs some thought and advocacy.
That's great. Okay, so we had a question with a couple more questions coming in. Um, so one of them is from Braxton Lewis. What biosecurity frameworks are being considered to effectively manage dual-use research concerns in synthetic biology?
Yeah. So great question. And as you can imagine, as with all technologies, right, there are a lot of, dual use possibilities. And I guess I'll say that it's absolutely an area that there's a lot of thought about, and you see it in a number of different ways. There are screening standards that are coming out where if you order DNA, it has to be screened. So if you're ordering something that's known to be hazardous, that it gets flagged and somebody says, wait, what are you doing? As you can imagine, there's the usual arms race, not quite that right term, but because now with generative AI, you're able to generate sequences that maybe have the same function but don't have sequence similarity. So how do we stay on top of that? And there was recently National Academy's report on AI and biology came out maybe 6 months ago that talks about that and talks about where that is. One thing to remember is on the biosecurity side, just in terms of AI and its impact, you still have to produce the stuff and you still have to disperse the stuff, right? So, what they said was on the design side of the design, build, test, learn cycle, that's where you're going to see a huge impact from AI, right? In the build side, maybe a little bit less, the test side, maybe a little bit less, right? So that's one thing to keep in mind. That's one. There are lots of discussions going around about, for example, dangerous gain of function research, right? This isn't necessarily just synthetic biology, but to some extent, all synthetic biology is gain of function in the sense that you're trying to get the system to do something that it didn't naturally do. That's the whole point. And so I think it was good when that came out that they were very clear about dangerous gain of function and what, trying to find what that means, put that in a little box. Because if you're trying to make vanillin, right, that's a gain of function, right? Yeast doesn't normally produce vanillin, but I can make it do that. You don't want that to suddenly get slowed down. So there is a robust biosecurity discussion. There's a lot going on, both in the unclassified and classified sides of things. So that's the good news. My opinion is this is an area where you have to have a kind of run faster strategy, right? I mean, this stuff is pretty easy to do. It is not hard to get biology. How hard is it to brew beer? You can do that right in your backyard, right? Or in your garage or in your basement. And so, it's not hard to go do these things. And so the way to be prepared is to say, we're ready to identify that something's maybe been released or getting used or maybe getting developed, right? And do something about that, right? Antidotes and other things. And so I think you have to just, stay ahead of all of that. Well, like AI, right? It's going to move fast. People are going to use it. So we just have to stay ahead on that curve and not try and, the US can do the things that we often do, say, oh, we're going to, put this under ITAR, we're going to do this other things. guess what? We are not the only country, right, who's moving forward, right? And I picked two, the UK and China, and they are moving forward very fast, right? And they are very aggressive about this because there's economic value to it. So I do think it's something where we're just going to have to run faster, stay ahead, and know what's coming and be prepared to do something about it if we see it. Biological weapons, I don't worry about. There's a, sort of biological weapons convention says we're not going to deal with those. I think people are going to abide by that. Biology makes a, kind of not great weapon. It's not 100%. It's not, always effective. It's hard to direct where you want it. So, but there are absolutely biosecurity concerns. They're absolutely getting discussed. I think they're important. They need to go alongside with that kind of run faster strategy.
That's great. We had a follow-up question for your point you made earlier in the presentation about sensors.
Yep.
So we have one question which asks, could you just say a little bit more about that one? That's not an intuitive application for biology. That's certainly when my ears perked up at, because I found that to be fascinating. So yeah, if you could take a minute just to talk more about the sensors applications.
Yeah, what's amazing about sensors is that if you have a molecule, so it depends what you want to sense, right? So let's start with molecules, but you can sense light, physical forces, other things, right? Think about what your body senses. It senses all of those things. That means biology can do that. But if you think about, molecules as a start, chemicals, we've gotten very good at being able to design a protein that binds a given chemical. And David Baker, who won the Nobel Prize a couple of years back, right, will say, I can design a protein to do anything. You just tell me what you want it to do, right? And Francis Arnold will say, I can evolve a protein to do anything, right? You just tell me what you want it to do. And so, I think the way to think about it is if there's a molecule, small molecule, DNA, other things, it's probably possible to build something biological that can latch onto that and detect it, right? And then do something with it. And the do something can be remember that I saw it. The do something can be tell you that I saw it. So turn some color like the hyperspectral images. So I think the thing that I would take away on the sensor side is anything that is sort of biologically detectable, and anything that affects biology is sort of by definition biologically detectable, right? And so light, heat, forces, chemicals, right? All of those things can be detected. And now we want to think about where can you put those things, right? And how do you get them there? And so again, in that hyperspectral imaging, you say, geez, what if I had a bunch of synthetic bacteria and I embed in those synthetic biology the ability to detect lots of different things and then have different absorption spectra, by producing a chemical depending on what they do, I can probably tell you a lot about what's going on chemically, right, in some environment. So I think it's a great area. It's an area where things like generative AI are going to make a huge difference as well.
That's great. Okay, well, we're just about at time, so I think we're going to have to leave it there. Thank you so much, Dr. Murray. This was great.
Great. Absolutely. Thank you for having me again, Sam, and for all of the help in organizing this, and to the NDIA for hosting this.
Outstanding. Well, thanks everybody for your participation. Thanks for the great questions. If you have any feedback for us, please feel free to send us an email at eti@ndia.org. I also want to take this opportunity just to plug our future webinars, our Tech 101 webinars. You'll see one coming through for November 2025. Be on the lookout for that one. And of course, if you'd like to register for these events, just visit our website or sign up for our mailing list. And there's a list of our upcoming Tech 101s and webinars. So thanks everybody for attending. Have a great week.